It's my pleasure to be here again today, and really in my 14 years I don't think we have been in such an exciting place with all the ice bucket excitement and all the awareness around ALS. And I really feel both the science, which has really at this time become so promising, and all of this influx and awareness, it couldn't be a better place for all of us today. So it's very exciting. Can everyone hear me clearly? Good, good. So what I'm going to try and do today, and of course there's so much to tell you and it's always hard to pick what are the best things, but I think because of all the ice bucket excitement, I thought I would really try and focus my attention a little bit about how we've spent the dollars. I've left with you also, and if those of you who haven't got, I've got more copies in the front, a little bit of a description of those programs and some other things that might be interesting. It is on our website, but sometimes it's nice to have it to hand and you can take a look at it. If you need an extra copy for a colleague or friend, I have a few extra here. So just really, as I'm going to describe, just a little bit about ALS. For many of you in the room, ALS is very much, well, for every one of you in the room, ALS is a part of your life, and many of you know about the disease. The few pointers I'm going to make is really to help us think about the science and how to think about developing therapies. And then most of the focus will be really the opportunities and challenges in developing therapies, but what an amazing time and chance we have now with the ice bucket money. So I hope I'll relay those points. So it's important to note a little bit about statistics, but actually when we think of it being a rare disease and affecting only 2 in 100,000, one thing you have to remember, and actually I say this often, it's only because on average people live a fairly short time that you have that kind of number, because actually if people lived as long as one does now with treatments with multiple sclerosis, you actually would have the same number of people. So when you think about ALS, it's as common as multiple sclerosis. The other thing to note is it's probably of similar incidence all over the world, although there are some peculiarities which we still haven't figured out. So that in the military, for example, there's a twofold increase in ALS. And in fact, in some populations in the past, in Guam, there was a 40-fold increase amongst the Camorra people. And we still puzzle, because it actually has now disappeared, the reason for that. And I'm not going to talk much more about environmental issues in my discussions, but in question time we can certainly have some more questions around that. I'm not sure I can have great answers for you, but I will tell you some of the thinkings and what we're doing. So 5 to 10% is thought to be familial. We were just talking a little bit about that concept, that you would have ALS in one generation moving to the next. Usually we think of it being familial when there are 2 cases in the family. Certainly these are things that you should be discussing with your neurologist if you have any concerns around that. 90% we therefore call sporadic, with not necessarily a clear cause, but also very likely to be genetic. One thing that has actually become much more realistic, though, is that figure of 5 to 10% might actually be a little bit higher, and it might vary within countries. And so there are some places in the world where that percentage of genetic is very high, in the Swedish, Finland populations, probably because there is one ancestral founder that is responsible for most of the ALS. And I'm going to talk a little bit about a very interesting mutation, which, where is my pointer? These I consider to be, and we as a field consider to be, the main mutations, or I'll tell you that there are many others, but these are the most prevalent and really track through families quite systematically. SOD was the first to be found. C9orf is the most recent of this list to be found, and actually 1 in 10 ALS patient is linked in with a C9orf mutation. So we actually know, at least for 1 in 10 cases, the potential cause of the disease. So that's a very different scenario than when we were a few years ago.
Can we interrupt you?
Yes, you may.
Go back. Okay, if you have that C9orf, does that make it familial?
So the question is, if you have C9orf72 as a mutation, is it considered familial? And this is a very complicated mutation. So I will perhaps readdress it later on again. The answer simply now we don't know. What I can say to you specifically, that C9orf accounts for 40% of familial cases where it tracks in families, and about 8% of sporadic cases. The challenge we don't know about the 8% sporadic is if they are truly sporadic. Is it because it's not a very penetrant mutation so that it skips families? Or, as I'll show you just now, C9orf causes ALS, it causes frontal temporal dementia, which is a cognition change, and it causes a combination of the two. So in the first family that we used to understand where that mutation was, it was a very large family and very curious because in some generations you didn't see the ALS, but actually you saw frontal temporal dementia and not ALS. So that maybe gives some of the explanation. So just for many of you who've seen my presentations, I often show this. It's only just to give you a sense of the biology and the challenge of the disease. It's not a disease in one single place. If you think of Alzheimer's disease, that's in the cells involved in memory in a very specific area of the brain. In ALS, our challenge is that it's cells called nerve cells, or motor neurons more specifically, that have their origins, the cell body, in the brain or in the spinal cord, and then are connected by these very long projections, our axons. They are one single projection that sends the message from the cell body all the way down to where it is connecting. And in fact, in this case, it connects from the upper motor neurons into the spinal cord, and from the spinal cord, it goes down to the muscles. And actually at that junction, that's what is required, the message all the way down from that initiating point all the way down to tell your muscles to contract. That's very simply what the biology is, and where the problem lies is a much more complex problem. Is it in the cell itself, in the brain or spinal cord? Is it actually at the junction of the muscle and the cell? Or is it in fact the surrounding cells? And what I started the field in 1993, I came into the field when we really thought most about the motor neurons, those nerve cells. And these are actually, in this case, human nerve cells, a very normal looking one, and one that shrivels up and looks very abnormal and has in it a very characteristic misaccumulation.
Is that in the brain?
This happens to be in the brain, but it can look exactly the same in the spinal cord. And that is a human tissue. We now know actually, and it has evolved over the last 20 years, that beyond the motor neuron, and I sort of promised you one single axon, but actually a motor neuron is much more complicated. It does have one single axon, which is the one projection, but it has around it many, many other projections, and all of these projections have to connect appropriately for the system to work. What we really do understand now is that it's not the nerve cell alone, but it's the surrounding cells, and that's an image of an astrocyte. These cells are very important in controlling the right environment for the nerve cell. They help regulate glutamate. Glutamate is a very normal component of the body and the brain in particular, because it's required for messaging, for sending round those proper signals, but too much of it can be a real problem. And it's this cell, the astrocyte, that is very involved in mopping up or removing excessive amounts of glutamate. And so if there's something wrong with the supporting cell, that astrocyte, just as much as the nerve being damaged, if the astrocyte is damaged, we might lead to problems with glutamate handling. To our right here, we have an image of a nerve cell. So where I said you have this one motor neuron, which in this case is traversing here, we have this motor neuron. It then gets to its end point, and this that you can't see so well is the muscle. And actually there isn't one single connection with the muscle, but there's what we kind of call a Christmas tree pattern. So there's a complicated connection at the muscle end. And in fact, this happens to be a mouse model in which you can do some clever engineering, and you can label in live time. You have that motor neurons labeled, so the animal is living, and you can look in live time at that connection. And that is what it looks like at its junction if it's healthy. If you take a mouse and you put a mutation in that's linked with ALS, you'll see that that all shrivels up and dies back. And all these little things on the Christmas tree pattern, these little dots here called buttons, so to speak, they are actually your chemical connection with that muscle. And they also have to all be very well connected for the muscle to contract. So it just gives you a little bit of a sense of how complex that system is, and that we have many angles to think about when we think of what's going wrong, and more importantly, how do we repair it. And then, as I was alluding to with your good question just now, ALS was always thought to be that problem of that nerve cell and the muscle cell, and clearly many of you are living with this challenge of the muscles being a problem. But there is also a component called frontal temporal lobar dementia, and it describes it exactly in terms of where the pathology is. It's in the temporal lobe of the brain. It's not Alzheimer's disease. It has, in fact, been confused with the two, and we only learned that because of the C9 discovery, and I can tell you more later about that. But it has certain behavioral and cognitive changes unique to frontotemporal dementia. And that's really important to know about, especially from the perspective of the family and of people looking after at the clinical centers, because that kind of behavioral change can also really show some personality changes that might be unique to that person and unusual because it didn't happen before. So now to move into a little bit of what we do at the association and my role in terms of what we run. And these were the strategies, and often I might talk about myself or we or I. All of it really is about, I lead it, but it's really a network, a global network of advisors and people that work with me who do all the advice. We have reviewers that do all the reviewing of things. So if I go into the we and I, it's really about a collective team of people that help think about these strategies. And the most important for me, coming actually at the time that I joined the association, I came from Bristol-Myers Squibb. So I was in the industry sector and I was very involved. My passion was to develop treatments for diseases. I had previously done my work in Don Cleveland's lab and developed a mouse model for ALS, so ALS was prominent in my interest for developing treatments. And so when I joined the association, my passion was to not only fund basic research, absolutely essential to understand how this is all happening, why is the cell dying, but to be able to translate those findings more effectively to get them to the clinic. So at that time, around 2004, after a bit more experience with the field and what was going on, I built the first translational program called Treat ALS for ALS. And it really was the first of its kind in the field, and also borrowed and working together with my colleagues in Huntington's and Alzheimer's and Parkinson's, who were at that same time doing similar things at a foundation level. So nothing has changed. That still remains fundamentally important. But what this new program, at that time new and now completely integrated into our thinking, could do was be able to fund initiatives that were really a partnership between the academic sector and the industry sector. And why is that so important? Well, probably to all of you, very obvious, but it's the industry that's going to develop the treatments. And the academics are truly the inspiration and the brains behind many of the discoveries. But as academics usually are, they will drift from one idea to another, and their focus is to do brilliant science, whereas an industry's focus is to be sure that they develop a treatment and to take it through a very systematic path. So having been able to build that partnership has been extremely effective and will certainly be something we will do a lot more of. The other thing we have been able to do through this program, and it's been really critical over the years, is to provide a much better infrastructure for clinical trials. And this is not to take away from the fact that we already have been doing and had been doing a great job at getting multidisciplinary care at the clinical centers. What I'm specifically talking about is running a clinical trial, which, because of the nature of the disease, where we need multiple patients in a trial, largely because we don't have effective biomarkers, which I'll touch on again. Big trials means participation from many, many centers. And to make sense of any of the data, we had to centralize it, we had to standardize it, we had to get everyone on board to be able to do the same measurements, to score the ALS-FRS in the same way, to do the breathing capture with FVC correctly. So that was a very, very big point of synergy that we were able to bring through our TREAT program. And then one of the things that has always been dear to my heart is really the concept that we never do this, even as a scientist, alone. You have to bring and build teams and teams of people, because I was at the time an expert more in molecular biology, and I could build a mouse model, but I knew nothing about disease pathology and how to analyze the data that I had in this mouse model. And I certainly am not a clinician and wouldn't understand all the clinical aspects that a person lives with and goes through. So bringing teams of talented people together is critical, and to bring global teams together, because it would be great if we find the answer here in Baton Rouge, but we may not. And so it might come from a collective effort between ourselves here and others somewhere else around the globe. And so that was my passion too. And having been, I did my thesis work, of course, in the UK, and knowing the teams there and having worked elsewhere in the globe, I think it really helps to be able to bring the different techniques and thinking in science together. So this is a complicated picture. You have it in front of you, and I think as we get more sophisticated, our picture will change. This was just in response to the ice bucket and to say, well, what is it that we can really do? And I spent the month, while others were dealing with various other things around ice buckets and media, etc. I spent at least a couple of weeks after I'd fielded many questions about stem cells and animal model studies, etc., which some didn't feel were right to do. I spent my time with my advisors thinking, what can we do that makes most sense of a wonderful influx of funding? And we didn't suddenly create programs. As I showed you, we had a strategic platform that was very solid. We actually had several plans in the pipeline, but never had the funding. And so this gave us a wonderful opportunity to energize around certain avenues that we had been exploring a long time. But central to this is the person living with the disease. And I think we need to look much more carefully at what the disease looks like in patients, as well as building the useful animal models and other model systems we have. I felt strongly that our 6 priority areas remain the same before Ice Bucket and after Ice Bucket. The science hasn't changed. So I felt that that was our strong basis to build on, our TREAT platform. And then I felt that there were real opportunities to become much more open source. So with this funding, let people have access to data, let scientists get more quickly tools that we need, and let people have access to analyze data in many ways. And again, part of this whole program and our research institute that we're building up are the clinical centers, absolutely critical, our local chapter work, and the public policy done both at national and at the chapter level. So these are the 6 topic areas, which again remain the same. It really helped us to focus our programs when we sort of decided that this is how we would describe the programs. It involves many complex different projects within each of the sections, but those are the main topics, and they're not really out of the box or terribly unusual. They are the key areas that we need to focus in and have been doing in many years. And I suppose here I just really highlight this point, and you'll see a fairly bare listing for environment. I put that there because really smoking is the only proven environmental exposure that's very well defined. The others are still lots of question marks. So there's a long laundry list for the environmental exposures, but most of them have question marks. Really, when we think about the disease, these are the components that are clearly happening when we think of the cause of disease. There are genes involved, and I showed you specifically the familial genes. There are environmental issues involved, which are here, and certainly in general, although we certainly see younger people with the disease, age is a major risk factor in the disease. So my first major focus then, when we think about our programs, is the excitement around genetics. This is where we were when I joined the field in 1993. I really joined it because of the discovery of this particular mutation in SOD1. And then it was rather bleak for a long time. For 10 years, we hadn't really found any other strong genes. We knew there were families, but we had trouble identifying the genes. And now the landscape is a very, very different place. You'll see many more genes than the 5 are isolated, partly because some of these are more risk factors. You don't see it strongly every generation, so they are a risk factor and probably have to go in combination with something else. A lot of them are genes in very, very small families, maybe even one single family. They haven't been able to find the gene in any other families. It makes it strongly a genetic factor in that family, but it hasn't gone beyond that family. And so the most exciting and important discovery was a C9orf mutation. And this was an investment for many, many, many years and millions of dollars on our part as well as many others. And it was a very, very elusive mutation that took a long time to figure out. First, we had the challenge that the mutation seemed to skip generations, but actually we realized then that, in fact, this mutation accounts for 40% of frontal temporal dementia. So what is very interesting, we have now a gene mutation that biologically connects to very different conditions. The damage in frontotemporal dementia is completely different to the damage in ALS. One is a nerve cell, the other is cells involved in behavior and memory. We don't understand why those biologies are like that, but of course that's why everyone is working in this field, because the fascination is enormous and the discovery of that is a solution to a lot of problems. These are the other genes, and it gives you perspective. So for familial ALS, we can account for approximately 60% of the causes of familial ALS, so a lot more healthy position to be in than in 1993. In fact, we think that the rest of the mutations to be found are going to be lots and lots of single small mutations in small families, maybe even smaller than TDP43, which accounts for 4% of familial ALS. SOD is 20% of familial ALS, and C9orf is 40% of familial ALS. But these are all very important clues to us, and why do I say that?
Is Parkinson's in one of those?
Parkinson's, this is talking only about ALS, but we can come to that. Only ALS, familial ALS, okay? And so why is that important? Well, this slide just shows you the impact of the increasing number of genes, but also to say that each of these genes helps us understand the biology, we hope and we think, provides a potential target for therapy, and helps us to build systems that will now tease out and question what kind of treatment is effective. So it really has helped us understand the disease better. So these are the 4 initiatives, which each of them I'll talk a little bit more about. The New York Genome Center, I've talked a lot about genetics, so I'll tell you why that center is so important. Project MIND, the Neurocollaborative, and ALS Act. So this is the New York Genome Center, and I had the pleasure for the first time of going to the center just a couple of weeks ago. And what this is that in some lead investigators, Tom Maniatis, who was the advisor to our scientific programs for many years, and colleagues in New York, felt it really important to build a center. So it's a completely new building. It's a beautiful building in the southern region of New York near Greenwich Village. They felt it important to be able to do all the sequencing of all the diseases under one roof. And so the center, in fact, does that, and it took on their advisory board all the New York laboratories, all the advisors in the vicinity to help them set that up. They now have the most current sequencing technologies, enormous machines, and very, very fancy equipment, the best of the best. The only other place in the world at the moment who has a similar setup is the Broad Institute, a similar idea in Boston. And so the idea being that now actually sequencing someone's genetics is a lot cheaper than 10 years ago and a lot faster. You can do this really, really at fast pace. So you might ask, well, why are all these genes emerging now? What is the difference between 1993 and now? In large part, technology and cost. So this seemed to us a fantastic, I knew about it, I knew it was happening, and I had said, you know, if there's something we can do and get ALS as part of this, let's do it. Of course, when the ice bucket money came, it was an obvious opportunity to say, this is for all diseases, let's make sure we carve out an area for ALS. And so that's what our first investment has done. We were able to get match dollars from a donor and the state to put $7.5 million as a starting seed to make sure that this would be a center where not only in New York but all over the globe people could go and bring their data, the DNA samples, for sequencing. They don't only do that part of it. I don't have the image here, but there's also an image. This is the wet labs, and this is where all the sequencing is. The second floor of the building is just an entire suite of people that sit behind computers, because actually what you need to do once you've sequenced is you've got to get all the information out of those sequences. And so these are all young specialists in the field in bioinformatics, churning through data and figuring out what it means. They are going to build into the system as well biologists and other people that when we discover what these things might mean, we test them in all kinds of biological systems. So a fantastic opportunity to be part of that. Project Mine was also a program that we had been asked to partner with, and we had in June absolutely no funds left to do any of that, so we said we would be the partner in kind, but we really didn't have any funding. We would start to think about the program, and when budget came along, we would invest in this. So it was a fantastic opportunity. I'll explain what it is, but our lead colleagues here are John Landers, he's a geneticist, and Jonathan Glass at Emory. He's a clinician who actually also runs the neural stem trial, if people are familiar with the neural stem trial. And to our right, we have Josh at Accenture, who really helped build the IT for this program, and his brother, unfortunately, also has ALS, so his company now are committing everything that they can through their employees into Project Mine. So what is it and why is it important? So Project Mine is a global initiative. This is why that's so exciting. It's going to incorporate many different countries. What is important in trying to find genes in sporadic ALS, so this is mainly the 90% that we haven't found the genes in yet, we need large numbers, because if you imagine each one of us sitting here has a completely different genetics that are very subtle changes. There are variations in every single one of us. Which of the variations in a person living with the disease actually causes the disease? And you can only do that by large numbers, by big comparisons, etc. So the goal is to increase two big studies, and the one that I've focused on for discussion is called whole genome sequencing. It's the new buzz in the genetic world. We used to talk about exome sequencing. Exomes are those regions in our genetics that produce or encode for the protein, and the whole genome is everything in between. So what's really surprising in terms of biological fact is that there's a very little bit of our genome that produces protein. Most of our genome is regulatory, but lo and behold, C9orf72 and other things are not found in regions where a protein is made. They are found in the regulatory regions. So we now realize with exome sequencing, we would never have found the C9 mutation, and maybe we are missing other mutations. So that's why it used to be far too expensive and prohibitive. We couldn't do whole genome sequencing. Now with the technology, we can do it, and of course our funds come at just the right time for that. And these are all our international partners. Everyone is involved around the world. And why that is so important, I reminded you to say that the genetics is very variable around the world. And in fact, in some areas, there are pockets of specific mutations. In the Netherlands, you will not find the SOD1 mutation. And in Sicily, you will find a large number of C9 mutations. So there's huge variability, and that's why we need global partners to figure that out. And the nice thing is on our website now, you'll see the whole story about our ambassadors to the program and the opportunity to do fundraising around it and to see if you want to sponsor one gene or two genes. It really engages the community in a very nice way to know where their money is going and what they can do to it. And so we've had whole programs a year ago, in fact, even less than a year ago. They had a swim in the Netherlands. In fact, this is where it started. It was a person with ALS, Bernard Muller, that started the whole idea of Project Mine. An entrepreneur who said it's crazy that we haven't got all the genetics about every patient with ALS. And so they had a swim in the channels in the Netherlands and Amsterdam. I don't think a very clean swim, but it happened. And there's one in planning for New York. I'm not sure what a New York swim is like, but we'll see. So then we go to the Neuro Collaborative, also a big business plan that was in the making before the Ice Bucket funds. And this was driven also actually by a person living with the disease in California who said there's such a wealth of intellect in California, certainly a wealth financially. We really need to do something major for ALS in California. And so conceptually there was that idea out there, and then I said, well, actually we have three very good teams. Why don't we try and put a business plan together around these three laboratories and see if we can make something happen? Well, we did write the business plan, and we couldn't make very much happen financially. So then came the Ice Bucket funds. And so we were able to get this program off the ground, and it has many components. And I think the area, and it will give me a chance to update you on the science, that's very exciting. Don Cleveland's lab, really through our TREAT program, was able to drive antisense, a very interesting technology, not only for ALS into the clinic, which it did go for SOD1, but for as a technique for SMA, for myotonic dystrophy, for Huntington's disease, for Alzheimer's disease, and now Parkinson's. And the first trial has gone on now with the SMA. Children have been injected with this antisense oligonucleotide, and it looks very safe. So this is to say that technologies are incredible, and we can impact not only our own disease, but many other diseases. And that's why we focused in on more efforts in that area. Our team is going to be doing something very novel in stem cells, and I'll show you that. And we also think that what's very important, although antisense is a way we can deliver something, and it's one technique, gene therapy is also an area that we need to focus on because it's also a very good delivery system. And so this team who've been able to drive antisense into the clinic will work with the gene therapy technique to get gene therapy into the clinic. And I feel very confident that if we can continue to support that, we can get that going. And then I'll talk a little bit about the iPS cell lines. So this is the antisense technology, and this is why it was so incredibly exciting at the time. And in fact, we built our translational programs in part for this very reason, because Don came to me and said, well, we have a crazy risky idea. No one is willing to fund it because we don't have much preliminary data. Would you take it on board and see if it could get into something for the clinic? And so we did, after peer reviewing a very simple, small proposal, we seeded the first grant at $200,000 and then $400,000. And then lo and behold, the concept did in fact work. And what it was is this company, ISIS Pharmaceuticals, they have an unfortunate name, but ISIS Pharmaceuticals, they basically had been doing this technology for local administration. So in retinal problems, and some work on stroke, but anywhere where you need to directly inject an antisense. They had never done it for somewhere where you have to get this traveling to get to your motor neuron. So the first simple experiment was a proof of principle to prove that if you put this into in this case, the brains of rats or mice, it would actually go to the neurons, and it did. It was remarkable. We would never have expected it, and it did. And that's the reason why we can use it for all these different diseases. And I think I have an easier image for you to see. So basically, in a bit of sort of 101 biology, here you have your gene. The gene then makes the messenger RNA. And the messenger RNA is normally what's going to make your proteins. But what happens is the antisense, which is a sort of strand of coding, it comes in, it connects with this part that is supposed to make a protein, and another enzyme comes in and chops it all away. So it basically digests and removes anything that was going to make a bad protein. Very simplistically said, okay? And then the intention is, if you get rid of that protein, hopefully then there won't be any disease. And it might have seemed a bit outrageous at the time, but proof of principle and studies going on is showing that yes, you can get rid of the protein. And as I say, it's in trial for spinal muscular atrophy, a baby childhood motor neuron disease. That disease is caused by one particular error in one gene, all of the cases. So you can see that this is a tremendous therapy that could maybe have benefit for every single person who has that disease. Not quite the same in ALS, but of course we think that we can find multiple targets. And SOD was the first, the poster child for this whole program. SOD is 2% of all of ALS, so it's only small, but it was a great proof of principle, and it's going to go back into trial, we hope, within the next year. It was in trial, was safe, but they've now got better chemistries to make a better system. But this is the C9orf gene, and you don't have to remember any of this, only to realize that where that orange triangle is, that's where the error occurs. Where the protein is made is over here. So just as I was saying to you, if we had done exon sequencing, we would have sequenced this and we would never have found this. And it is an insertion, so it's junk that comes in. Well, it's 6 hexanucleotide repeat that we have normally about 20, and people that have the C9 mutation and living with the disease have up to 1,000. So it's an expansion in a normally repeated domain. Why does it have such a horrible name? Because we don't know what the protein does, we don't know what it's doing. It's chromosome 9, which is where it's found, on an open reading frame number 72. So when we understand the gene, we will give it a name, but we don't understand the gene yet. And that is the remarkable thing. This was discovered in 2011. We don't know what it does, but the day that it was discovered, ALS Pharmaceuticals, Don Cleveland, and many others started to work out the treatment approach, because one approach might be, with antisense, is just get rid of that. We don't know what it does, we don't know where it's going, but we know we don't want it. And the good thing is, with antisense technology, and let me just click one more time here, this is just to show you what I said, and this is how we can measure it. So normally you look like this if you do a little blood test and a PCR, and when you have the expansion, you look like that. So basically, that's how you differentiate a normal and a control. It's a bit simplistic. There's some challenges with it, but in simplicity terms, that's how you can differentiate. But what is very exciting now is if you can block that production of this, you don't interfere with the normal protein, because remember I said to you that this is the bit that's going to give you the product you need. If you just block that, you still will get the normal product. So that's why it's so interesting that you can tackle it as a therapy. And that's about all I'm going to say about that. But the exciting thing is ALS Pharmaceuticals, who started out with a small support with us through Don Cleveland, and we probably invested maybe up to $500,000 to $1 million, is now partnered with Biogen. And Biogen is running all the very, very expensive trials for SMA, and we'll do all the trials for ALS, for SOD1 and C9. So it just shows that you can mobilize with a small investment an incredible investment downstream. And I don't know that number, but if you think that dexpramipexole, a much simpler trial, because it was just an easy pill administration, that cost us, the company $80 million. So you can imagine that these antisense trials are going to be extremely expensive. So this is just to tell you why we also chose to focus on gene therapy, and that's because actually not so long ago the field of gene therapy was kind of dead, and that happened because of a horrible toxic error and someone dying many years ago now from a gene therapy trial, a normal healthy individual. The other problem for our field was that the vectors in trial didn't work very well for us. And let me remind you, gene therapy is basically an approach where a viral vector, it's a virus, infects into the body and can go to, for example, the motor neuron. It infects, but it's not infective. So you won't get the virus like the flu virus or whatever. You won't get anything from it because it's what they call attenuated. They've blocked the infective part, but they've enabled to keep the ability for it to migrate to where we need. So it's purely just a delivery tool. And now we have much better delivery tools. We call them AAV9 and AAV10. And all you have to take home from this image here is that this is from a mouse. It's a brain. It's a spinal cord. And if you look here, this is the spinal cord. And in these three cases, you can see very little, in fact, no green lighting up. In these two cases, the whole spinal cord has been able to take up that virus. So that was essential for us to be able to develop gene therapy in ALS. And if we go to the next image, if I have it here, yes, the same is true for the brain. So this is the brain from a mouse or a rat, I'm not sure anymore. But anyway, this is the brain showing you with these kinds. Actually, in the brain, AAV8 wasn't too bad. But the 9 and 10, the new versions we've developed, are extremely effective to get in the brain. So from that basis, we can really start doing it for ALS. And in fact, I would say the partner to our collaboration, Brian Casper in Ohio, has just treated the first babies in the SMA successfully, at least for safety perspective. The babies were treated with these viral vectors, and he's now going to team up with us to do it for ALS. And that's why that was an important priority to fund. Then moving a little bit more quickly along, Steve Finkbeiner is the second lab from the Neuro Collaborative. He is very well known amongst the academic field for this technology, which is very exciting. He can, in an automated microscope, which nobody has to sit behind it or do anything, everything's set up automatically. He can shift through these plates of wells, which carry now these stem cells from patients, the iPS lines, and then that's why I wanted to show you it in this order. He can look at the cells over time because it's just taking pictures all the time of it growing and developing. This is a very healthy looking normal type of cell, but if you have cells from patients that have been developed from the skin cells that have abnormalities, you can pick that up, and then at the same time you can start putting drugs into those wells and say which of the drugs that we might be interested in are going to change the look of those cells and help them survive. Obviously, this is very much a crude early part of seeing if drugs work, because it's a cell in a dish, and it's certainly not a whole 3D system, but it gives us clues of what looks promising, and then we can go and do much more complicated studies. And this can be done so much more quickly to give us clues of the right things to test. And this is just to remind you, because I've used the term iPS several times. iPS is induced pluripotent stem cells, a really exciting technological discovery that you can take skin cells and then you can manipulate them. They used to be done with 4 genes that you manipulate. It's now become much more sophisticated, because in fact you can not only take skin cells, but you can take blood cells now and transform them into stem cells that make them then either the liver cells, heart cells, lung cells, and in our case, motor neurons, astrocytes, and muscle. Because what we're also doing in very exploratory studies is building a spinal cord, and motor neurons, astrocytes, and muscle into a 3D type of thing in a dish. So we're trying to kind of build humans in a dish in a very simplistic sense. And this is just data to show that from a C9 patient taking their skin cell, we see exactly what we see in humans. We see these dots accumulating in the cell, and we know that if we treat with antisense treatment, which is already in development, those dots go away. What that means yet, I don't know, but we have a readout at least to say when you do something, you can actually get a physical change. So you don't have to understand the complexity of the slide, but because of the importance of iPS cells and stem cells for the community, we decided to build a core at Cedars-Sinai where we will develop all the cells and all the biology around it, and then be able to, through a contract organization, send it out to the world in terms of what it is that they might need to use. So I'm going to move a little bit more quickly through the final thing called ALS Act. This was a very exciting opportunity. These are programs that were already starting to begin and had been funded by a single family donor. And I was less involved with it until the Ice Bucket money came along. And this particular donor said, well, we have these programs, and I knew all the players. And in fact, I didn't know that the donor had been a partner to us before on some clinical trials, because he had wished to remain anonymous. He then decided that he really wanted to do something big, because his daughter-in-law has ALS. And he said, if we put in 10 million into a program, will you match it with another 10 million? And so we now have a 20 million program, which is doing amazing things. Together with that partnership, General Electric was already a partner. General Electric Health Systems is very, very well known for their talent and skills in imaging markers. So in other words, being able to, in lifetime, look at changes in the brain and hopefully now changes in the spinal cord. And so with their technology, which they are also applying for brain trauma as a partnership with the NFL, because of the issue of brain trauma in players, we have this partnership with them and this foundation to really make things happen in a big way. So part of the program is going to be biomarker development, a critical tool for us. I think I have more slides to describe it. Therapeutic development, so there are a few exciting programs already in place, another gene therapy program using a slightly different approach, and a program looking at anti-inflammatories in a very different and unusual way. We have supporting what is Neurobank, and what is very exciting about that is now suddenly, now that we're investing so significantly in so many programs that are collecting patient samples, require clinical data, require blood, skin, and CSF, we are able to connect all those studies together and centralize them through this Neurobank. So first of all, there will be no doubling up of a patient. Even if they've gone to 2 places, we'll be able to, through a global identifier, connect their data and be sure no one's replicated. And we will be able to also standardize, like we did our clinical trial systems, we're going to standardize all data collection so that we now have the ability to have a really large data set which we can analyze much more effectively. And we have just put out a call for proposals to globally and also to industry to fund phase 2A clinical trials. These are just before the phase 3 studies, so they're smaller groups of people. But what we think is essential and that this call is asking for is that if we have a trial, you have to have a design that has together with that trial a biomarker. So we have to know at the end of that trial that the trial has failed because it's the wrong pathway or the drug doesn't work, but not because we don't know where the drug actually acted. And that is critical because we've had too many failed trials and we don't know how to analyze whether the pathway or the disease mechanism was wrong or did the drug even get there. And that's what is required for us to fund any of these studies. And then we are going to next year roll out a few different initiation of ideas and ask for people to come with proposals to this particular part of the program. So why biomarkers? For many of you, you know the frustration it took to get diagnosed. If you went to a specialist center, then you were probably very lucky, but many of you will have gone through other places first, maybe your general GP, maybe even to ear, eye, and throat, nose and throat, whatever it is. And so that pathway is far too long. We need improved diagnostics, preferably a blood test, but it might not be as easy as that. But we need to be able to build up earlier diagnosis. Very critical for clinical trials, because if the disease has progressed so far, the ability to affect and change that is much more difficult. We need to be able to think of people in a trial not as ALS as one disease, but be able to group them. And I say this because there might be trials just like in the cancers. If you carry a particular genetic mutation in cancer, you'd go for this kind of a treatment. And I think that our trials are failing because we put everyone in together. There might be some that respond better to anti-inflammatories because they have a profile that looks like they are prone to that kind of thing. So being able to segregate is very important. The one way we know how to do it is through genetics. So the next two big trials that will be on the horizon, we hope, are the C9 and SOD1 trials using the antisense technology. Well, of course, the patient population for that is going to be SOD carriers and C9 carriers because we know that's the target. But we hope we'll be able to learn how else to stratify trials according to certain profiles. The other area is that the reason partly why industry is very slow to embrace ALS as a disease in their portfolio is that the trials are exceptionally long. Most of them have to be done over 9 months to 2 years because we're looking at progression of disease using the ALS-FRS. ALS-FRS has been quite good for us, but it certainly is not quantitative, it's qualitative. And so the measures are not absolutely subjective. And also it has to be done over a long time frame. If we had something like in the MS world, where they have now a brain imaging marker and they can see a lesion, and the minute that the drug has an effect, that lesion changes, then you have very short windows of time that those experiments have to be done. And I know for sure there'll be many more companies wanting to be in the space. The other thing is what I mentioned to you earlier. We need what is called a pharmacodynamic biomarker, one that says that the target is engaged. For example, dexpramipexole or the EMPOWER study was thought to be working at the level of the mitochondria. So can we really say that that drug has got to the mitochondria and engaged with that target? We certainly couldn't in the EMPOWER study. So we have to build tools and expect people to do that. Most clinical trials and most industry studies do that, and that's how they move forward. So part of this then is going to be an effort to collect samples, which we've done and are going to do much more of. The other is a very busy slide, but to tell you what do samples do for us, well, you can then take blood or spinal cord, and in some cases we compare signatures from animal models and others, and you can start to bin these signatures into groups. This is an MS person with MS, and this is what their signature looks like in a normal, actually no, this is a healthy, HSC is a healthy control, and this is ALS. You start to see that people are segregated according to profiles. And so it's probably not going to be a single marker, but it's going to be a collection of things that say this is ALS versus this is a person who doesn't have the disease, and this is Alzheimer's or Parkinson's or something else. And that will help really in defining and help us also change how we think about trial design. This is a final slide about biomarkers, and this is a really exciting experiment that we've been funding over the years before we even had the influx of big money. And this is what I talked about, an imaging tool, and this is what General Electric can do. This is in a rat model. And the green is showing here a transporter that I talked about earlier. I remember I said to you that in the astrocytes, in those cells surrounding neurons, they were particularly good about removing too much glutamate. And they have a special transporter to do that. And that transporter is called EAT2. That transporter engages with glutamate and gets rid of it. We had many years ago a trial, ceftriaxone. Some of you might remember the trial. It had to go through an infusion, quite complicated. Some felt the trial might have worked. Well, certainly statistically the trial failed. But we didn't know whether it was because the trial was doing what it was supposed to do, which was affect this EAT2 transporter. If we'd had this imaging biomarker at the right time, we would have been able to see the changes in this particular transporter because of the drug, or not if it wasn't changing it. And more importantly, we can see in humans whether this transporter is even affected in the disease. Well, now this has been completely completed. The way to do it has been tested in the rat model, and it's working effectively. We've just paid the last parts of the toxicity testing that was required to be done in the rat model, and it's going to go into human clinical trial. The trial is going to be purely for the marker, so it's not testing a compound, but it will only need to be small, and it will really tell us that when we have drugs that are going to affect this kind of system, we will have a way to test in lifetime in humans if this is actually happening and if the drug is changing it. So this is the kind of thing we are going to invest more and more in. There are obviously wishes to have brain imaging. I'm going to skip. There's a lot of information. And this is also just to move from saying we'd love to have a good marker in the blood or the spinal fluid, but work done by one of our scientists is on this chair, the ALS. And it seems to be a very, very good system for measuring strength. And it's an adaptation that's been done over time, which is now testing in a small clinical trial and seems to be a much more comfortable and reliable measure of muscle strength. And this is something we are going to help her introduce into the steps. So what kind of things are happening next? We're going to develop a partnership with Jack's Laboratories for mouse model development. I'll tell you a little bit exciting about that. We are going to do many, many more academic industry partnerships because I think that's where our successes have come from, and more will come. We are going to do a variety of clinical studies and clinical management type studies. And we are going to continue to support our young investigators through our postdoctoral fellowship program, because not only do we need great ideas and equipment and materials, we need great people to do the work. And this is just really just to reiterate that our programs have always had this concept of funding from very early development all the way into helping people living with the disease and improved management of the disease. And one example where that was very successful was an early idea for the SOD antisense therapy, and we brought that all the way to clinical trials. So it can be done, and it will be done much more effectively over time. Model systems are very important to us. It tells us all about a very complex disease, and these are all pathways that we can think about for treatment. And this is just one last final fun piece of data. We recently had a big workshop where my colleague Kat presented. And mouse models have had a hard time in the press more recently, not only by the ethics folks who feel that we shouldn't be using mice, but that's an aside. They do carry their leather handbags, so I don't know, that is a bit tricky to then put the two together. But the more important thing is the challenge, does a mouse really reflect the human disease? Well, a mouse is not a human. We can't make them both the same. And I still have a strong bias that the mouse has a very important role to play. It will help us when we develop treatments because it has a blood-brain barrier. It is three-dimensional, and as fantastic we can be in a dish with our motor neurons and our muscles and our rest, it still isn't the same system as a mouse running around. So I do believe it's going to be an important tool if used in conjunction with our stem cell lines, our human information, that we can much better understand and integrate the things. In fact, we also have a dog model of ALS, which has the SOD1 mutation, and I haven't got time to talk about. So mouse models are important. Not everyone else will tell you the same story, but I believe that is the case. And this is just to tell you the reality of clinical trials generally not only for ALS, but for every disease. How long it takes to get down that pipeline, how many failures we get from 50% of things looking good all the way down, then we get in an animal model and it looks pretty good, and it goes to phase 1, and then 90% of the things fail. So we've got to get that system better, and that's not a problem that I'm thinking about alone. The industry is frustrated by it because in reality they are not doing very well at finding new drugs, especially in the neurological space. It's so tricky. And the mouse has a good role to play, and this is a fantastic piece of data actually, just to say that we have to get better design, etc. So when I was in Don's lab building mouse models, it took me years to get the systems going and to create the mouse model. In fact, new technology called CRISPR technology, and this is to show you what it means, it doesn't matter, in 8 weeks you can have a mouse model. So it's gone from really almost a year to develop a mouse model to 8 weeks. So that's a phenomenal technology change, and we're going to capitalize on that. We're going to help build for the community many more models. We're going to own them so that we don't have all the issues of legal tech transfer from academic centers, etc., so that companies can use them. And we're going to build a resource where we have multiple of these genetically modified mice, genes linked to the human disease in mice, to start to understand how these all compare and how similar or different they really are. So I think with more tools we'll make a huge impact. So this is just, I thought, a cute slide of hers to say technology has made an enormous impact and we have to use it well. And so we, I think, with our new Ice Bucket funds can capitalize on that particular opportunity. And the details of that are not so important, really. I will skip forward to tell you the challenges of drug discovery are not unique to ALS. Billions are spent in the world, and this is an old time point, but really billions, and the products and the number of drugs coming out are very, very minimal. And so we can really be the glue between the academia and the industry to help really move things along, to help with the difficult parts of model building, getting the right patients into clinical trials. There are a lot of things that we can really do, and that's where our funding is going to make a big impact. We do have the industry interested. We have many companies involved in ALS today. Many always feel frustrated, well, ALS is a disease that nobody knows about. Well, the Ice Bucket changed that. The other is that companies, in fact, I get an email a day from a company that has an idea and wants to work with us. They haven't promised money yet, but they want to work with us. And so this is only a snippet. Biogen IDEC is on the top of the list because they actually have invested an enormous amount. And most companies, after a failed trial, they drop off the scene. And Biogen is here for a while, and so that's a fantastic thing. They've funded big academic adventures into exon sequencing. They are very, very committed, so they will continue to do so. Some other big names, Pfizer, GSK, I don't even think is on my list. They are intimately involved with ALS studies. Not funding big studies yet, but very much at the table to discuss and help provide service in kind. So many of these companies, if they have a good academic partner, they will provide even an employee or materials that are needed, so at least they are giving something back in the partnership. And finally, to end on the young people, because these are our fellows. There are many older and more senior people like myself in the business, but these are the young folks through our fellowship program that really are going to be the drivers. There are a small representation of them. Many of them have actually now become independent and have their own labs, and they are really the scientists for now and in the future. So I think I covered pretty much everything, and I hope I haven't exhausted you all. So I'm open for any questions if you have the energy for them.
Great presentation. So you've given us an overview of a whole lot of complex research that's going on and given us numbers as to what part of the money is going to different grants and this and that. Do you have a big picture out of that roughly $100 million, whatever it is, that's going directly to research? Is there a way that y'all have summarized it?
So there is, I mean, there's been a lot of discussion within the organization, and I think that there's, I have to repeat the question. The question here is, so how are we from the 115 million allocating funds, and how much of that is to research? And so the easiest answer to tell you is that for the pure research projects and programs that I've showed you here, the bulk of that money is going into it. I think it probably would come around about 65 to 70%. The reality is, though, it's much bigger than that, what is going to impact research, because through, excuse me, through public policy efforts, we have a program to do work with the FDA. We are thinking about an FDA fellowship program, so other things that are really actually going to be directly impacting. In the clinical arena, there's going to be much more support for clinical centers. We are going to work to see how we can improve the clinical components that are part of our program. So there'll be other monies that go to more peripheral things and not directly into the research programs that I describe. I hope that's helpful.
I just recently stopped taking Rotec. What is your opinion on when drug therapy doesn't benefit patients? Have you ever seen sea lion off the coast of South Africa?
I love those questions. I've got to repeat them. So the one question was that you were on riluzole and you've gone off riluzole. And what do I think of drugs that don't really work, what you should be doing as such? And have I seen sea lions off the coast of South Africa? The last question is the easiest. Yes, I have. You go to Seal Island, which is actually close to when Mandela was unfortunately in prison. It's a very smelly journey, but yes, there are sea lions there, so I've seen them. Thank you for the question. So in terms of riluzole or Rilutec, I think there's variable enthusiasm for the drug. Some people feel that it does work for them. What is real is that in combination with things, all these things might add up to be helpful. But if it is, in your case, not working, or you feel that uncomfortable with side effects, etc., then there's absolutely no reason, without good consultation with your clinician and what he thinks as well, that you might decide to stop it. So in the UK, most people are on riluzole and stay on riluzole unless they have a side effect, because it's covered through the medical system there, and that's pretty much everyone does it. Sometimes it's the enthusiasm of the clinician as to how enthusiastic the clinician might be about riluzole. It has a modest effect, if it has an effect in some cases, and that's where I say it's very possible that there are some people that have more of a benefit than others, and we kind of lump everyone together, but it might be very variable. So no, if the drug is not very effective for you and you don't feel a benefit, that's a decision that you make together.
I just recently stopped taking Rilutec. What is your opinion on when drug therapy?
I hope that answered it. If you have more, then no problem, ask again. And you know, sometimes when they have modest effects, it is very difficult to know. And what we hope to do is maybe with modest effects and this cocktail approach, that maybe those things will enhance the ultimate effect.
Where
Yes.
are you getting your samples, like from people in Louisiana where we don't have a clinical, any clinical trials going on or anything? So if you want to give a skin sample or a blood sample and get it in this big mix, how are you going to do that?
Okay, the question is, so if you wanted to be part of a study, and if you're here in Baton Rouge, and how do we get samples from people that are excited here into our bigger programs? So I don't have a quick answer to that. We are working on it. So because these 4 programs intersect in such an exciting way, a lot of the work from the ALS Act initiative is going to be sequenced at the New York Genome Center. A lot of the samples that are collected are going to be developed into IPS lines at Cedars-Sinai. That's exactly what's going on at the moment. I'm going to be identifying how we're going to connect them together. Certainly the Neurobank is going to do that. Which are the current centers involved? Where are centers that could be added? And then finally, when we've got it all sorted out and we know what clinical data we need and how we want to do it, all the standard operating procedures, which is not completely newly being invented. They are around, but they are in lots of different places. Once we're able to embrace and centralize at least how we do it through a spoken hub type system, we're going to have a very effective area on our website that will exactly say who is participating, which centers, where you can go, what you can do. So it's all evolving to bring, and at least hope to bring, as many people interested in getting engaged in the program engaged. Does that answer your question?
What's your timeline?
Well, the timeline is in the next couple of months. We are finalizing all the contracts and getting things rolling and getting the first collection started. The timeline in having our website up and about, we hope in the first part of next year.
Okay. But you can't give blood like in Baton Rouge and have your neurologist send it to.
Absolutely. Absolutely you can.
Okay, what are you doing to encourage the neurologist to be involved in this?
So the question is, can someone send the sample from here to somewhere else? Absolutely. These samples can all be sent. What are we doing to engage and incentivize centers? We are going to, once we've got our ducks in a row, because we obviously have to have the system first set up. We have to get the buy-in that we've got the right clinical questionnaires we are going to be questioning, and then we are going to see who would like to be engaged and how it would operate.
And if I could add, it also is dependent upon your encouragement of your physician to participate and for them to ask questions as to how they can be engaged. I assure you we're doing quite a bit to engage neurologists and other physicians in Louisiana and Mississippi, but your impact that you can make in speaking to them about their involvement is very important. Part of the peripheral funding that Lucie mentioned is related to greater establishments of our clinics and getting our clinics that are involved. As you know, the ALS Association Louisiana-Mississippi chapter has one clinic in which they are currently supporting, and that is at Our Lady of Lourdes in Lafayette. We are also looking at ways to partner with other operations in the New Orleans area, and also trying very, very much to bring something of the same operation here in Baton Rouge. So again, your voice is very important in helping us to bring this to fruition. And so please continue to encourage the physicians that you are seeing to be a part of the growth of the ALS Association and helping to engage you in clinical trials.
Thank you, Kelly. That was excellent. Yep. Other questions?
Yeah, what happens when y'all run out of money?
What happens when ALSA runs out of money? Well, I hope that we will not be in that position. And just to then really clarify again, in terms of the spending, obviously there was a lot of pressure that we spend it all, we don't do endowments. Well, in reality, even just talking about the 4 big contracts that I'm working with at the moment, it takes time to do the best thing with them. And so that is a first initial investment. We will do that well, and we have already 10 million as a partner donor. We have 5 million in the New York Genome Center as a partner, so we've already leveraged beyond what we got as the Ice Bucket money, and that is my intention to do more of that. We have a sort of 3-year plan with the rolling out the Ice Bucket money over the 3 years. We hope to entice more revenues around that time so that it might even last longer. And we are very much spending in research on a milestone-driven basis. So if a project looks very exciting, we will give them the investment to get it going. If it's promising, we'll give them more investment. And the way especially we'll do the drug contract programs, which is what we did with Antisense, with ISIS and Don Cleveland, is you have lots of different programs to begin with at maybe $500,000 or less each. And then we see which of those are going to be the ones that are successful, like a funnel effect. And we'll eventually take a few of those all the way through. But just as perspective, it takes about $1 billion to develop one treatment. So our 100 million is a fantastic start, but it's a leveraging start. If we can do lots of these things with real potential and drive them to the clinic, I do believe we'll get the partners coming in. If we can build a fantastic sample collection with excellent clinical data, with imaging tools that General Electric will help us develop, as others will do as well, we can go to the industry and say, we have this platform, we've invested X amount into the platform. You need biomarkers if you're going to do a trial for ALS. We need you to come and be part of the team. And we hope that they'll come with their dollars. So we have ideas of how to continue to leverage, but we really do feel that everyone should recognize this is not enough, and we need to keep putting more into it.
Question, I know with the FDA, it's harder or it takes longer to get drugs to market in the U.S. So as far as these studies that would be done here, are there more roadblocks than there would be in the UK or in
So the question is about FDA and the roadblocks and the length of time it takes. A little bit of that is not quite accurate. In fact, sometimes trials are done here because it is more tedious in Europe because you're dealing with the EMA that has 10 different countries and languages and all kinds of complications. So, and the FDA does have many programs like the Orphan Disease Act, which is to help expedite the various PDUFA acts, which are really focusing and bringing in ALS as a partner to them to explain the risk-benefit effect. So there are lots of things ongoing. Things are being expedited, and I think often the bottleneck is not so much with the FDA. In some cases, yes, and those are things we're talking about, especially with things like antisense technologies, gene therapy, stem cell therapy. Those three areas which are very new in this space for any neurodegenerative disease, where safety is a real concern for them because, you know, gene therapy, the field did shut down because of a death in that field. So those are more tricky fields, and you know, maybe there are ways we can do it better because now each stem cell trial is requiring each of them to do more or less the same initial safety studies, etc. So we are working with that. But I think in essence, FDA has always been, in the last while that I've been working with them, quite a good partner and very open to listening. And here and there where things are not quite so reasonable, we can have a huge impact. So that's why part of the investment that we talked about from the ICE bucket will be to work with FDA and to really see how we can improve systems.
If you're eligible for a
Is there a
trial,
national biomarker bank setup?
I'll answer your question quickly because you might have another question you'll need to line up, and then I'll answer yours. So is there a national biobank, right?
Biomarker.
Oh, a national biomarker. Okay, that's different to a biobank. So you might mean both, so I'll give an answer to both. A biomarker, internationally, yes, ALS-FRS, FVC, muscle strength measures. Those are the international ones. Survival is still the most approved marker for clinical trials and most liked in Europe because they feel ALS-FRS is not quantitative and survival is a clear point and measure. So those are internationally accepted and used in clinical trials, and it's what was used in Empower study. There is, however, is there a national biomarker repository? No, there is not. The CDC, the National Registry that all of you are familiar with, is trying to work on a biosample that will go with every single person in the registry. It's a very complicated area to have a national repository around, and we feel that initiative is good and it should continue, but we think with our opportunity we are able to, in a way, collect and connect so many things where we have to be sure the clinical data is important, the ability to go back to that sample and that family and say we want to see it over time versus a one-time commitment. So there's so much that goes into a biobank. I think to do one huge national one connected with the registry is going to be a challenge. Not impossible, but maybe we can do something very effective with what we have. So I gave you two answers, and I hope those are helpful to you. Repeat your question, please.
If today you wanted to get into a study, which ones are most promising?
So what are the most promising studies to get into at the moment? So we are in an unusual time where there are very few phase 3 studies. So Empower was the last very big one we were hopeful about. I do know that some of you are familiar with the cytokinetic study, which is in phase 2, which we hope will go to phase 3, and that was to help really with physical strength. That might roll out into phase 3, but there aren't many phase 3 studies at the moment, is the honest truth. We are hoping to get a lot more excitement around phase 2 studies. If we can fund a few of those that are really good studies, they might be the ones to get involved in, but there are not many big phase 3.
Question on studies. The familial version is kind of what we think we're facing. And I noticed on the website that there was something about the University of Miami, and I don't understand phase this and phase that. Are you familiar with that one or any other familial?
Yes, so for familial ALS, what studies are ongoing and what is the study with Michael Benneter at University of Miami? So Michael Benneter has been funded by us for quite some time, and what he is, and he's a very good contact who I would be happy to get you in contact with, or Brian Traynor, some great colleagues. They are focused at the moment on SOD1 and C9, and they are broadening out a study, and what they do is we call them natural history studies. Basically what they are doing is they're looking at families that carry, for example, the SOD1 mutation. You can be part of the study, have the genetics done, but not know if you're a carrier, so you can just be part of that study. And then what is good about it is that he's collecting biomarkers, blood, CSF, probably skin samples by now, a whole variety of things, and doing imaging, and hoping that when he does it completely blinded, you look at all the different markers in whether it's control or someone carrying the gene, that eventually we can say long before anyone shows a physical signs of ALS, be it speech or in the limbs, we can see a signature that's going to give us a clue. And more importantly, how early before diagnosis does that signature appear? Because a very important question that we have no answer to, and you kind of alluded to it, you carry around, in some cases, a genetic mutation from birth. And in most cases, it really is midlife before that particular mutation is a problem. It probably is that we all undergo wear and tear, and that nerve cell is an incredibly unique cell in that it has a very big demand to do a function, getting messages along that long distance. You put in something else, and on top of that something else, all those things are eventually going to sit on that wear and tear system where we would maybe age, and we do lose nerve cells going down as we age, but someone with that on top of it, this kind of mutation, suddenly the system can't cope. That very much is likely what is happening and is why you manifest so late. But that said, how early are you seeing this? And very early studies from Michael's work are quite fascinating in that actually the change in signature that he's seeing is only quite close to the time, at least the markers he's looked at, and there might be other things he doesn't know about yet, but the things he knows about that are changing, they seem to change quite close in the time frame before the disease, which is a very fascinating and remarkable thing, because at the time you get the disease, your decline in number of motor neurons have dropped already quite dramatically. So something is suddenly happening very fast. Or we're missing something. I mean, we don't know everything, and so we still have to figure that out. In Huntington's disease, a study called HD Predict, they are able to define almost in the early years of life that there are changes going on in brain volume. And so it's so early that these changes are happening. And that is extremely helpful then for them to start to design at what point is it so bad that we can see a clue and we must think about a therapy. Because, for example, if we have antisense SOD1 and it really is a working, every therapy, as we know, has side effects and problems. We don't see anything major yet with the antisense, but we haven't given it long-term to anyone. So when are we going to start giving a family that thinks they're going to show ALS in 10 years' time, if we can predict that, is that when we're going to start antisense therapy? So it's all those kinds of questions, and that's exactly the kind of studies that are going on with Michael Benneter. And he's a great person to connect with amongst others I could connect you with.
If you don't have familial ALS, and I'm not sure I have the language to ask this question, but if you don't have familial ALS, is there a way to know what kind of genetic mutation you carry? You have to be involved in one of these studies to know what your
Okay, so
specific issue is?
good question. If you don't have genetic ALS, or you don't think that your ALS is running through a family, and you are considered sporadic, how will you know which genes are involved? Well, I'll tell you, even in familial, because there's 60% of the familial genes unknown. So, for example, your family could decide to have a blood test, and you say, well, we'll look for SOD1, C9, VCP, TDP43. None of those are positive, but you certainly still have a gene in your family. We just don't know it yet. And so in those cases, we can do a test and say that you don't have all these genes, but you still might have another gene. In sporadic, it's much more difficult, and that's why we're doing the study like Project Mine. We don't know the genes. We do know that in some cases, very rare cases, SOD1 is seen in sporadic, very rare. We see 8% C9 in sporadic. We sometimes see TDP43, so some of the genes in familial have appeared in what seemed to be a sporadic case. But it's so rare that if you have sporadic ALS and is not tracking in the family, generally speaking, a clinician and a genetic counselor will say there's no reason for you to have a genetic test. We might get closer to understanding what to do when we get the results from something like Project Mine, where we're trying to collect all this information and see the variability of genes. The problem is in sporadic is you might have a gene, but you might never get the disease, because otherwise you would not have been considered sporadic. You might need 3 genes to get the disease. You might need 3 genes together with something that's fundamentally very important in the environment. So it's a combination. It's what we call the genetic susceptibility, is your combination of genes together with something else that causes ALS. So very, very tricky to know it.
If you have, if you're not sure whether you have familial or sporadic, is genetic testing warranted?
No. So is it warranted if you're not sure if it's familial or genetic? Well, if you have a family history of 2 or more people in the family, I highly recommend you speak to a clinician who is well versed in this area. If you don't have any family history, there's absolutely no reason to get a genetic test done.
What if you have family history of, say, ALS and someone else had FTD?
So if you have a situation where in your family you're sure you had frontal temporal dementia and ALS, I would highly recommend you speak to your neurologist and discuss it with them.
Well, how would you even be sure if you've had dementia in the family in past relatives that are deceased? You wouldn't know if it's FTD or not, but does that warrant being tested? But in the case where you don't know if it's FTD or it was dementia from Alzheimer's, what would you do? I would certainly speak to your neurologist. I mean, there might be value for you to look at the C9 mutation and be involved in a study there. And again, Brian Traynor and colleagues would be the people to connect with. I think that if there's clearly been Alzheimer's disease in the family and they clearly say it's Alzheimer's, I wouldn't be concerned. If you're unsure of the dementia, it's worth a follow-up discussion. Another question. You or somebody else had mentioned on one of the YouTube videos that most likely therapies would include multiple treatments, multiple therapies. Right now, the clinical trials are only approved, but they don't admit you to a clinical trial if you've already had another therapy, right?
So the question is, if multiple therapy is a way to go, why are all the treatments at the moment just single therapy? So I will say that if you do a trial in Europe, and in fact the dexpramipexole trial was set up this way, in Europe you can't do a trial without riluzole because everyone is on riluzole. So you will have an arm of riluzole and dexpramipexole, and you'll have an arm of just dexpramipexole. So that is quite acceptable because riluzole is a standard of care in the UK, for example. And I think that that's how we will get multiple therapies. It's very difficult to be sure if any one drug is doing anything, at least at a small level, if you don't have, you'd have to, unfortunately in a disease like ALS where our trials are so big, the only way that you could comfortably do a proper design trial would be to do exactly that. Your single, your double, your triple, and you can then imagine how many arms you're going to have to prove that. Because you cannot really throw in a cocktail and say which one was the one that worked, because we can't then further develop that any one compound that was effective. So that is how it will do. It will be building one up on top of the other.
Could you tell us how the EPA program is going to work?
One moment, and then I'll get back to you.
Have they done anything to figure out why military people have a 60% chance, better chance of getting it?
So the question is, why is it there an increase in the military of ALS? And so we have done several studies where we have collected DNAs. We have looked at some potentially interesting genes involved in dealing with and handling chemicals, pesticides, that kind of thing. The PON, it's called the PON 1 and 2 and 3 system, and I can't quite think of the acronym now. It's the end of a long discussion. I can get back to you on that, but the long and short of it is we're unclear. We continue to try and collect. It's very interesting because it's a defined population. It is deployed and undeployed, so that's kind of the interesting part of it. Not everyone is necessarily exposed to the same thing. And so the theory that colleagues have had, and some good colleagues in the UK sometimes talk about, is that, you know, what is the connection? Is it actually just the physical, healthy strength of the individual? Because we're seeing it in sports, we're seeing it in marathon runners, we're seeing it in soccer players, and what is common between all these people is that they're actually quite healthy, physical, athletic, strong people. And so it sounds a bit of a kind of ridiculous, and we have no proof of it. But it's like saying, well, one model is a Fiat car and the other is a Mercedes, and somehow the Mercedes has that architecture that's more vulnerable to something. That is what some geneticists have said, that maybe there is just a genetic makeup that goes with that physical nature that unfortunately in some other way is sensitive to the injury. Nothing proven, but just an idea.
The EPA program.
So the expanded access program is really something that's driven from by the FDA. And that is in phase 2, it's almost a bit compassionate access to treatments. So I think that program is a good program. There's discussion around it, and companies can be embraced by that. Certainly Biogen was talking closely with the ALS Association that should the treatment be effective, that they expedite getting it. The challenge at the phase 2 level is sometimes that, you know, these are small studies. So in fact, in Biogen, it seemed like at phase 2 it was going to work. In phase 3, it didn't work. But there certainly are discussions, and it is very much dependent on whoever's running the trial as to whether this is something that might be able to be offered. So it's a complicated area. It depends entirely also on how easy it is to produce that particular treatment approach. For example, for stem cells, it's not so simple. Because it's an invasive, very invasive treatment, and there's no real proof yet that it's going to be of benefit. For things like antisense, it's not going to be trivial to develop the therapy. So it is very variable, but certainly we are working in any way we can to give access where it's reasonable, or where at the moment there are not many of those examples going on because there are not many successful phase 2 trials that are pushing us to think about that.
You said that there was very definite personality changes in people with the frontal lobe dementia and or ALS. What do you mean by that?
So the question is, what are the kinds of personality and behavioral changes that come with frontal temporal dementia? Again, not being the clinician.
Link it to ALS.
Oh, linked to ALS. Well, it will be so it will, if the ALS and FTD come together, the FTD is the same as an FTD alone. But obviously in FTD alone, they don't have the motor symptoms. In ALS, you have the FTD, and there are sometimes just mood changes, temperament changes, unusual social behaviors. I name a few of the things that I've learned along the way, but there are really good summaries on our website and clinicians that really focus in on frontal temporal dementia. There's certain executive learning word association studies that are problematic for FTD. So there is a battery being developed of testing that can be done if there are issues around that.
Okay, let me give me the...
I will connect you in with that, and also if you have any questions, Dr. Kathy Lohman-Hirth is one example of, she's a great clinician who's really focused her energy around FTD. So I prefer that they give you a better description of it than I probably have done.
Are there any defined groups of people, whether it be ethnic or geographical, or even as specific as groups of people who have cancer or another disease, that are where ALS is completely non-existent?
That's a very good question. Usually people ask it the other way around, that there's an association. And so generally in population-based studies, they have not seen protection or a lack of it in one the question really was to say, is there a population or a series of diseases like cancer that would then exclude you from having ALS? And I think in reality, it seems to affect most populations. They have not, in population studies that have been done, it doesn't seem to show a spared population. You know, the early registry studies, seemed to indicate it was more prevalent in a Caucasian population, but I think in reality the studies are too early to make that statement very boldly, and I think many others have done studies where there isn't a great difference. There will be pockets where you do get it. In Tunisia, there's because of intermarriage and through families, there's one recessive gene that has been dominant in that area, so there's an unusual occurrence of it in there. So there's more pockets that are unusual than that are protected that we know of at this time. But they are very good questions and actually not impossible to get by if you start to look through health databases.
The last I've heard of clinical trial, you have to be, once you're diagnosed, you have 2 years that you're eligible for a clinical trial. It sounds like with the new technology and research that there may be more clinical trials in the future. Is there any chance of extending that timeline to where if a person's diagnosed 3 or 4 years that they can still get into a clinical trial?
So the question really is about eligibility for clinical trials and really the urgency and the need for a person to have had been closer to diagnosis than later. And the reality is that as clinicians and obviously as people developing treatments, you want to have the best chance of the treatment succeeding. Because you obviously, if that happens, then it goes out to market and you can get many people can benefit from it. So in many cases, the concern is that the disease has progressed too far to have a benefit. That said, however, if we get better biomarkers for the disease, I think there's much less of a problem around that, because then you wouldn't be so concerned that you would have slow progressors that would skew the points in the clinical trial. You would have a clear biomarker of what's happening and not use the progression of disease as your biomarker, because ALS-FRS is really a measure of progression. And so that's why it's so difficult. The other is that there might be some forms, for example, the cytokinetics trial, which is looking at muscle strength and is using FVC as a big measure. It doesn't really need to exclude, as long as you're able to see a change within your own profile. It need not exclude people that are slower in progression. So it will definitely depend on what the treatment is, but the reason for it often requiring it to be much closer to diagnosis is you want to hedge your bets in the best way to make sure something succeeds. And that's where the compassionate use, the expanded access becomes so important, because of course you do want to see if others can get access that couldn't in the trial.
You talked about ceftriaxone. Are they starting to open that study up again?
The ceftriaxone trial, will it open up again? No, there really at this point is no reason to. Unless we really get a very good marker. For example, I'm only saying, no one said this, but I'm just thinking aloud. If our E2 PET marker works really well, it wouldn't be an unreasonable compound to put in a very small trial, just as a proof of principle. But I know of no such study planned.
Does the association investigate claims of reversal or cure from these trials?
Does the association investigate cure or reversal in trials? Well, yes, they do, because we're doing exactly that for one particular situation. So many of you know Ted Harada, and I am amazed myself at how well he is, at least when we last saw each other. And he was in the neural stem trial and had stem cell therapy. And it seems highly unlikely, and the investigators will attest to that as well, that it was a stem cell therapy that had the effect. And so the clinicians involved came to the association with a proposal that maybe it was the immunosuppressant cocktail, a number of compounds being used, that might have been
what did it. And so now we have a small pilot trial which is looking at the no stem cell therapy, no invasive therapy, but just the immunosuppressants. So we don't know. It's maybe a fishing expedition, but it was worth an investment to give it a try.
What about the brainstorm occasion?
Okay, so the question is, what about the brainstorm study where there was one indication, at least I only know of one and there might be others, and unfortunately there, and this is what media does and press does, he was misdiagnosed. So he had a treatable disease that had some signs that looked motor neuron-like.
What anti-inflammatories are you talking about? Any that are on the market today?
They are a combination, a cocktail combination, and I can give you the exact names at a later point, but they are always used for tissue rejection. So the reason why they're given is in a transplant where you're getting stem cells that are foreign to you, and you would then obviously need to have it. So it's like, it's really the very usual combination given for transplant therapy.
Which makes you susceptible to pneumonia and a lot of other things.
Well, that's why that actually was one of the things that they had to look at very carefully from a safety perspective, and they obviously had to get the dosing right, and if anything, that's the harder part of the trial rather than the stem cell transplants. But so far, and of course the trial is ongoing, and that's carefully monitored. It's not ideal, of course, but it's an opportunity if something works.
What about people getting stem cells overseas, a lot in Mexico, Honduras? I mean, for some reason.
They're all.
So I think the one thing I would be saying about if you're going to go and look into stem cell therapy somewhere, do check their website for a few things which I always find alarming when I'm highlighted about a stem cell therapy. The stem cell, the treatment that they give treats every single disease. If you look at their scientific advisors or people that are on the scientific team, I'd be hard-pressed to find a specialist in our area. They've often come from completely different fields, maybe veterinarian or something else. So be suspicious of that. And so be very careful about it. You know, I've had less calls now than I did before because we have some trials that are FDA approved. But, you know, there are people that have had to change their whole lifestyle because of a stem cell trial, meaning that they might mortgage their homes, they might do all kinds of things that really has an enormous impact. And so far, I've not heard of any of those trials being successful. Sometimes people have died going to those trials, not because of the trial probably, but because they didn't have their same care around them, or that's how the disease progressed. But I would be very cautious of stem cell trials that are not really carefully done with approvals. If the team there are not willing to speak to some of the experts or other clinicians or your clinician, I would be suspicious. So I'd be very careful of that. All the stem cell trials, even if they are here and they FDA approved, be it Brainstorm, be it NeuralStem, be it others coming, they are experiments. These ones here are carefully done experiments, but they're just experiments.
Were there any benefit from nutritional treatments like vitamin D or?
So the question is, what about nutrition and the benefits from vitamins? Well, again, I think there are studies out there to try and work that out. Vitamin E was particularly one of them because SOD1 fits into that pathway as a free radical scavenger, etc. I think it's always where the approach or the vitamins are safe and it's good to take, gosh, it can't do any harm. In terms of nutrition is incredibly important, and I'm probably not the expert to talk about it. I know that it's part of just care. It's absolutely important. And there are certainly very interesting studies about high-fat diets and whether actually that is good and people benefit from that more so than not. And so we are about to support some studies in that area. We do think that diet is an incredibly important part, and so we are, through our clinical management program, very responsive to those kind of questions.
And I'll just piggyback on that to say that if you aren't seeing a dietitian during your disease, and you are, I would highly recommend that. And the one way in which you can do that is through your care at a multidisciplinary clinic. And one of the things that I wanted to share with you all today is that the announcement was made today in regards to the ALS Multidisciplinary Clinic at Our Lady of Lourdes in Lafayette, that beginning at the December clinic, we will have Dr. Fady Malik, who will be joining the clinic as pulmonologist. And that will then put us at a fully staffed clinic, whereas you can seek your treatment, and we hope that you will do so. Very important to also add, and something that Lucie very nicely alluded to, the registry. If you have not joined the ALS registry, it's very important that you do so. It is one of the most important research projects that we currently have going on. And there are many ways in which you can have impact in this disease if you are registered through the registry. So I encourage you to speak to your care services person about that. They're certainly available to help you register if that is something that you need assistance with.
And I'll piggyback onto that. One of the things from a research perspective that the registry can do, and why you would really want to be involved because of your good question about which clinical trials, what clinicians and scientists can do if they have a specific project and a question they want to ask, they can send a request to the CDC, to the registry, to ask whether or not the registry participants would like to be involved in a study. And so I believe when you have the form to fill in, there is a question there that says, would you like to be contacted for any other clinical trials or studies? And so technically what happens, a researcher sends their proposal to CDC. They want to engage a population with maybe a specific description. And then the CDC, not the researcher, emails out to those people that seem to fit that particular study, and then they get the contact details of the researcher. So for that reason, it can be very valuable to be part of the system so that there might be an opportunity to be in studies that are coming up.
Excellent point. Is the clinic in Lafayette currently thinking about joining some studies or not, or what? Are they anywhere?
The question was, is the clinic in Lafayette considering joining studies? Yes, that is not completely and totally up to the personnel that are there. It takes dollars for them to do that, which one of the things that Dr. Bruijn was discussing was how those peripheral dollars will be supporting the clinics around the country and engaging those that are currently not involved in trial studies to possibly become a center for that.
I'm sorry.
Do you find that
Can you talk to us a little bit about Lunison? Am I saying it right?
Lunison.
It's when I have it on my phone.
I doubt whether I can because I don't recognize it, but I'll try.
What about drugs are being used for MS? Are they any good for ALS?
What about drugs being used for MS, for ALS? So you're absolutely correct. There is some exploration of that. In fact, there is a study going on that ALS TDI is doing, and I'm blanking on the name of the Galenia, thank you very much. It's been a long day. Galenia, and in fact, that is the concept that maybe because of the inflammation component, it might have value. Galenia has some side effects, so it's being done very carefully to see if it is a safe treatment, but it's purely because it potentially could work on in the inflammation part.
And not be an anti-inflammatory with all the other.
We don't know that yet, but yes.
Okay, it's Lunasin.
Lunasin.
ALS and Fingal was talking about that at the Arizona conference.
I didn't hear what he spoke about, and so to be honest, I don't know. I would have to look into it a bit more.
Okay. Elaine?
One little question that my husband Jim has been diagnosed for a year or so, and he asked if I would ask you if, and you sort of have answered this, I apologize, I know you're tired, but if you had ALS, what direction would you pursue? he initially said, would you be trying to get into a stem cell study? But I hear what you're saying, you know, I think your answer would be not necessarily. But so then what direction would you
What
take?
direction? Okay, so it's a tough question, and I'll say the question again. So if you're newly diagnosed or if you've just had ALS for a year, what should you be doing? What can you participate in? What trial? So definitely engage with the registry, really engage with questioning your neurologist, what's out there, what's going on. There are, as I say, there are phase 2 trials coming up. There are some phase 2 trials, so I would urge your husband to look at our website, which says clinical trials. Okay, and if you click onto clinical trials, it will link you into the NEALS site, which has a listing of the trials. He'll probably be completely confused and say, well, what must I do with that? He has there a number and an email address for my colleague who really works through any trials that might be of interest. It's on the website. Okay. Perry is her name, but all her contacts, email, number on the website from clinical trials. I would certainly start exploring tissue involvement, being involved in sample studies, collections, because that would be great. And as I say, they're sort of organizing themselves, and there's no time frame for that. There's no, if it takes a couple of months for us to sort out this, and he can participate at any time, for example. Getting involved with the registry.
Contacting your local ALS Association, Louisiana-Mississippi chapter.
Pay political announcement.
That's his favorite thing to do. Good, thank you so much.
Okay.
Since the Ice Bucket Challenge and y'all have all this money, do you find that there's the same sense of urgency among the scientific or research?
I would say, so is there a sense of urgency? I would say there's a huge amount of excitement around the Ice Bucket funding. So just a few weeks ago, I had already planned it a year ago, but 120 of our scientists in all fields, clinicians, basic scientists, all came together to share their data. And the energy was phenomenal. And the reality that they could talk about projects with new ideas and how we could collaborate, knowing that there was real potential to fund those ideas, was phenomenal. So I think the energy is huge, and I think that the opportunity is amazing. So I don't think that anyone was less committed before the Ice Bucket Challenge, but partly because of the way we constructed the first big programs, there's a lot more realizing that we have to figure it out to do a major project and all link in somehow, and much more collaborative thinking is going on, because everyone actually, even in the investigator community, everyone feels they have an ice bucket in front of them with money. I mean, everyone is, even though they're not all going to get it in the same way, they all feel engaged with that academic exercise of how would you spend ice bucket money. And so from that particular workshop, what I had the fortune to quickly plan, knowing the ice bucket money had come in, I had 12 topic tables around various things. What are the gaps? What do we need for a biomarker? What are the limitations of clinical trials? All these various areas, and each of those teams were tasked to brainstorm around them and come back with their ideas. And so I've collated all those ideas, and I'll tell you, a lot of that is going to be the driving force of how we think about rolling out new solicitations for proposals. So it's been a phenomenal energy amongst the investigators. And also remember that the association received funds, but in Italy they received more funds at the ALS community there. The MNDA received more funds. So globally, people have received more funds, and we are talking together to figure out how we can partner around those opportunities.
Do y'all use expediters to try to expedite things and keep the schedules?
So there was a question that you'll have to repeat before I can repeat it. Say again.
Do y'all use expediters to coordinate the schedules and make sure that everybody has the same sense of urgency?
I'm not sure what expediters are. Maybe that's a program I'm not aware of, but...
No, he wants to know, does the ALS Association pay anyone to keep everyone on track?
Oh, okay. So That's my job, although I'm pleased to say I'm getting one additional staff person because my job has evolved to becoming enormous. But beyond that, absolutely, to coordinate all these things, that is my role. And I have teams of people that are responsible for various things, not my teams, but the investigators and clinicians. But everyone has to be kept to task, and you're absolutely right, that's going to be a big job.
Well, that's from the patient's perspective. I mean, that's the main job, getting the therapy on the market.
Yep. But money goes a long way, and then money in a clever direction goes a long way too. So that's exactly what we're working towards.
I feel very enthusiastic to think that you're the expediter, because you certainly do convey a sense of real clarity and your ability to articulate all of these different issues.
Sweet. Thank you for your kind comment. Thank you. That's very kind.
Thank you. One more question, Chrissy.
Actually, not a question, it's a statement.
Okay.
Just because everybody's talking about the Ice Bucket Challenge. I don't know if anybody else has seen it yet, but Time Magazine has Pete Freights up, which is the guy who initially started the Ice Bucket Challenge. They have him up against, some Ebola doctors and all, for Person of the Year.
Fantastic. Wow. Very, very exciting. So we're really excited to hear that Pete Freights, he's got the year of the, what is Time year of the Time Magazine nominee for, okay, Man of the Year, Person of the Year. Thank you. The team reset that question statement for me. That's fantastic.
Does the ALS Association have ideas for another? What are we going to do next year?
So will there be another ice bucket? Everyone always asks that. And of course, it would be fantastic and phenomenal. But I think, so there'll be lots of activity around the Ice Bucket Month, I'm sure. And that's something that our development teams and communications teams and the chapters will brainstorm and work around. I think that there's a huge opportunity to leverage funds. And certainly my role, I hope, will be to communicate some great success. It's a big challenge to do it in a short time frame because science takes long, but I'm hoping we'll have some very good talking points around things that have been achieved.
There's 2 movies coming out.
That's right.
Steve Hawkins movie and You're Not You.
So there are 2 films on ALS, which is very exciting. The Steve Hawkins, I saw the trailer in one of my hotel stays along my 10-day journey recently. So yes, it's coming out. Very exciting. I think the attention is phenomenal, and I don't think that will suddenly disappear. I think that it will stick and very much is in everyone's minds, and it's brought people that we would never have thought of in a much more long-term commitment with ALS. So I think those things are phenomenal. Very exciting.
We have time for one more question.
Thank you. It's been very interesting, well presented. Thanks for
my pleasure.
Come down and share with us.
My pleasure. I'm very pleased, and I know there's much more information to come. And actually, I do encourage, for those of you who have not been involved or haven't had a chance to, we try and hold every month a research webinar. I've been a little bit slow at getting them off the ground over the summer because it was just so crazy. We have actually the next one, so you might want to skip that because you've heard it all, is going to be me talking about the ice bucket, very much what I've told you here today for callers from all over the world. But then after that, we're going to roll out lead scientists on each of these 4 projects to talk about their progress. So do encourage you. It's 30 minutes of the scientists speaking. I try my best to get scientists who can speak clearly, and if they don't, I remind them and I try and correct them and help make it easier. But it's just fun to hear from the actual people who are doing the work. And then we have 30 minutes of Q&As. And actually what's really nice to see is that many of you come, but actually scientists also come into the call because they're also curious. So it's quite a diverse audience, but it's meant for all of you to feel on top of what's going on and welcome to suggestions of topics as well.
I'm just wondering, some of the slides that you kind of went through quickly.
You can have the whole set.
We'll be glad to share that with you.
Okay.
And they will
For
also...
my engineer.
Yes.
I mean, he would love to, yeah.
Absolutely, we will have the slides. They'll also be available through the YouTube piece.
Okay.
So
Yeah.
we'll
I
have-
didn't know.
Yeah, I would ask just keep it with yourselves and keep it as a PDF if you can, only because sometimes a slide is rather meaningless without my presentation with it. It will mean something to you, but I don't want people to interpret things incorrectly because we haven't talked about it. So it's for your reference, would be perfect.
All right. Well, thank you all very much. If you have continued questions around the webinars that Lucy was talking about, your care services person has that information for you that can share with you as to when those are happening. And we encourage you to be in touch with them related to especially those events, because Lucy does very much keep the research folks to task regarding their presentation and our ability to understand what they're saying. So please make it a point to participate in that. Again, thank you very much for coming.
Thank you all. It's great to have you.